Assalamualaikum. In this video, we will be presenting about the study of methionine analog-based cell-specific proteomics and interactomics. MACSPI enables tissue-selective proteomic and interactomic analysis in multicellular organisms. So the overview of this video is we will talk about the introduction of the study, principles and applications, also the advantages and disadvantages. So I am Nur Amira Shahida, will be talk about the first part of this video. The chosen articles related to the study of proteomic where it is significantly connected to proteomics since it used the MACSPI approach to identify and quantify proteins in tissue-specific context. Other than that, the article also related to interactomics by identifying tissue-specific protein-protein interactions, and the proteomic data obtained from this study can indirectly benefit the metabolomic studies. And lastly, the study relates with the system biology by combining proteomic and interactomic data to create a comprehensive picture of tissue-specific biological processes. For research objective, the study focuses to evaluate MACSPI method for tissue-specific proteome and interactomic profiling in multicellular organisms, also to illustrate the ability of MACSPI in detecting protein complexes and interaction in certain tissues. The other objective is to study the tissue-specific interactomics and proteomes in the C. elegans multicellular model by applying the MACSPI method, and also to identify unknown tissue-enriched protein and relationships by comparing tissue-specific proteomic data acquired by MACSPI with whole organism datasets. Method used in this study are transgenic C. elegans strains, stable isotope labeling by amino acid, and scilab profiling of TOC-specific HSP90 interactors. So this is the workflow of the experiment where the method used genetically modified C. elegans to study protein near HSP90, and worms were feed labeled bacteria and treated with a special chemical to mark nearby proteins. These proteins were then collected and identified using mass spectrometry to find HSP90 interactors.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Najihah binti Azhar, and I will continue this presentation with the key findings of the research and conclusion of the research. So the key findings of the research first is tissue-specific proteome profiling, which the MACSPI technique allow precise isolation and identification of proteins from specific tissues within a whole organism rather than the homogeneous samples. This will overcome the major limitation of the previous proteomic studies where tissue context was often lost. For example, in C. elegans, MACSPI enabled mapping of the neuronal versus muscle-specific proteomes, which distinguish proteins unique to each tissue. The second point is the detection of protein-protein interactions, PPIs, with spatial context, where MACSPI integrates proximity labeling, such as TurboID or BioID, which tissue-specific expression system, which enable researchers to capture protein-protein interactions in their native spatial environment. This is revealed the distinct interactomes in different tissues, which are essential to understand the functional protein networks that vary by cell types. And the third one is dynamic and condition-responsive interaction mapping, which MACSPI detected changes in the interaction networks of the protein involved in cell signalings or stress responses, which shows the method's dynamic of tracking biological processes in vivo. And this method can capture protein interactions under different conditions or even in the developmental stages. Continue with the conclusion of the research, which MACSPI is a novel and effective method for tissue-specific proteomic and interactomic analysis in multicellular organisms, which it allows precise identification of proteins and both of the interaction partners within specific tissues while maintaining their native biological context. MACSPI technique is robust, sensitive, and minimizes cross-tissue contamination, which improve the accuracy of the proteomic studies. It is demonstrated successfully in C. elegans, showing its capability in live whole organism beyond cell cultures. And MACSPI enables the study of dynamic, condition-dependent protein interactions, making it valuable for understanding biological processes and disease mechanisms. MACSPI opens new avenues in developmental biology, neuroscience, and biomedical research, which has promising future for use in higher organisms. Overall, MACSPI is a powerful tool for dissecting complex protein networks with spatial and functional resolution in vivo.
Hello, my name is Aina. So I will continue my part that is about principles and application of this topic. I will be start with the principles of this topic within the method that have been used is methionine analog-based cell-specific proteomics and interactomics that can be short formed to the MACSPI. There are several of the principles of this topic. The most important principle that have been used for this method is Tissue elegans or C. elegans. It is a modern multicellular organism for identification of tissue-specific protein which can be functioned to express tag proteins. The second primer is engineered methionine aciditase or METRS. It is an enzyme that changes methionine A which non-natural amino acids photoannounce into proteins. The third primer is photoactivatable non-canonical amino acid, which can be short-formed into photoannounce. It is an analog of natural amino acid that contain photo-cross-linkable group. This analog is functioned for site-specifically into newly synthesized protein in living cell. Tissue-specific promoter in C. elegans also play an important primer for this method, which can be used for drive MACRS expression with photo-NR in targeted tissue. The last primer is UV light exposure. It is used for photo-cross-linkable group in the photoannounce to form covalent bonds to protein of interest. Next, we go to the application of MACSPI. The first application is tissue-specific proteome profiling. It can be helped in understand cell type specific protein expression by using MACSPI. Next, mapping protein-protein interaction or interactomics also can be applied via MACSPI, which allow in vivo analysis of protein-protein interaction in a tissue specific. The third application is drug discovery and disease research. It can be used to facilitate the discovery of tissue-specific therapeutic targets and help in uncover mechanisms of tissue-specific pathologies. The last application is validation of protein function and localization. It is used for protein targeting, identifying mechanisms, and functional annotation of uncharacterized proteins.
Hi, I'm Mizati Wafika. Will present about advantage and disadvantages. First thing first, let's talk about the advantages of MACSPI. It is considered pariah methods. In order to support MACSPI's advantages, the author contrasted with PRIO-ID, Turbo-ID, and transloctome profiling. Next is tissue-specific precision. This method allows for the investigation of protein and interactions in specific tissues found in multicellular organisms. This method also have low background noise. MACSPI show a greater signal-to-noise ratio than previous labeling techniques. As for the disadvantages, MACSPI requires genetic modification. This is depending on turbo-ID fused protein transgenic expression, which might not be possible in all species. Other than that, it is variable labeling efficiency. The reproducibility may be impacted by variation in biotinylation performance among tissues or circumstances. Lastly, it is limited to organisms with genetic tools. This is because it is less effective in environments with weak transgenesis technique. So, that's all from us. Thank you for watching.